Okay, so if you guys take a look at this picture, it has three different parts to it, right? So it has, this is, they call the ribbon model. This is not showing, the blue is showing, is supposed to be the phosphates and the sugars. And then the bases are in the middle and it's showing the hydrogen bonds. And the point of this model is to show you the double helix, that there are, this is the B form of DNA where there's a, what we call a major groove and then a minor groove. So yeah, this is wider from turn to turn, and then here it's narrower. This is the chemistry model, right? And this part of it shows each of the nucleotides. So remember, a nucleotide has a nitrogenous base, sugar, and a phosphate. In this case, the sugar is deoxyribose. So here's thymine, which is a pyrimidine. And notice that it's paired with purine with A, right? And A always pairs with T, and G always pairs with C. That's the complementary part, the complementary base pairing. And then notice that each of the nucleotides, so the nitrogenous base is always bound to the sugar molecule, which is in blue. It's a monosaccharide, and like I said, it's deoxyribose. And then there's this phosphate sticking out here. The sugars bind sugar, phosphate, sugar, phosphate, sugar, phosphate, and the bases bind to the sugars. Now, the important piece that I didn't really stress very well on Wednesday, I think, is that you see how on this end, we call it the 5' end. And what that means is that there is a phosphate attached to the 5th carbon of this sugar and to the 3rd carbon. So if we go all the way back to chapter 5, where we counted carbons on glucose, right? We start counting after the oxygen. So this would be the 1st carbon, 2nd, 3rd carbon has a phosphate. 4th carbon, and the 5th carbon is this one right here that you can see. That one is also attached to a phosphate. Why is this important? Because the other end of the DNA molecule of this particular strand, there isn't a phosphate here. There's a hydroxyl group. And so we call this the 3' end. Now, the complementary strand, we say, runs in the opposite direction, right? This isn't 3' here, it's 3' 5'. That's what we mean by anti-parallel, okay? Structurally, the sugars have to flip upside down, but this is how it's paired, okay? So on the left-hand side, we've got a 5' to 3', and the complementary strand, we say, is anti-parallel or runs the opposite direction, 3' to 5'. The other last important sort of chemistry part to note is that between A's and T's, there's 2 hydrogen bonds, and between G's and C's, there's 3, okay? And one way to remember that A pairs with T and G pairs with C, since we live where we live, I always tell students, if you think about at Gloucester City, right, then you can't mess it up. Okay, I wanted to show you that, and then I think I'm going to draw the replication and go through those 7 steps with you. Get rid of all this nonsense and go back to I'm stuck. How do I do that? Stop the sharing. There we go. Minimize, make me bigger. Okay, so now I was going to real quickly go through the replication with you, and I typed out those notes for you guys and has the 7 steps, and the 7 steps match the picture in your book with the 7 steps. I did that on purpose. In your book, it's on page 326. And I'm just going to go through those steps with you and try to do a drawing here on the shared screen on the whiteboard, because I don't know what happened to the video on my computer, but it doesn't seem to work as well as it did before. Okay, so here's I'll start out in black. Here's a piece of double-stranded DNA. So I'm going to draw this just like I would normally on the chalkboard in class, right, like that. And this end, when you write a molecule of DNA on horizontally like this, so this way the 5' strand is always on the top. When you write it vertically, the 5' to 3' strand is always on the left-hand side, like it was in the book. So I'm going to draw the complementary strand here. And this part that I'm drawing right now, this represents the phosphates and the sugars, right? So I'm going to put a 3 here, a 5 here, or 3' and 5'. And then if I draw the lines like this in between, those represent the bases, right? So like one of these lines would represent, I don't know that I can draw that sideways, but like if there was an A right here, right? The complement to that would be a T over here. G, C, whatever they are, right? So I'm just going to draw a few more of those. Okay, the important part is the part out here where the where the helix has been unwound and unzipped. And so I'm just going to make the bases a little bit shorter here. Right. So the molecule that starts this, that does this, we call it unwinding and unzipping because it's going to separate the two strands of the DNA. That molecule has a specific name, and that molecule's name is DNA helicase. And I'm going to draw it kind of as a cube here and put it right there. I need a thicker line for that. Right? And I'm going to kind of color it in. Yeah, that's pretty good. Right, so the green blob is DNA helicase. HELICASE. ASE, remember, because it's an enzyme, right? And its job is to break these hydrogen bonds. And that's wonderful, except if you break the hydrogen bonds and you don't put something there to keep them apart, as soon as that big enzyme moves a little bit out of the way, their molecules, the hydrogen bonds will just reform and the double helix will twist back up. So there are these proteins that sit here just during replication, and they're called single-stranded binding proteins. And they kind of look like U-shaped clamps because they actually, like if my arm's the DNA, they actually sit on the DNA like this. Let's make them a little thicker too. They're hard to see. So we usually draw them kind of like that, or sometimes books will just draw them as like little circles because they cover up the, they hold the DNA open. So I'm going to put a line out for them, and I'm just going to shorthand call them SSBP because single-stranded binding proteins is way too long for me to try to write right-handed when I'm left-handed. Okay, so helicase unwinds and unzips. Single-stranded binding proteins, they're going to hold that replication fork open. And now the enzymes that actually do the work of copying the DNA can actually fit in there. And so I'm going to make DNA polymerase red. And DNA polymerase can only read the DNA. So it can only read this top strand. It can only read 3 to 5, and it can only add complementary bases in an anti-parallel way. So it can only add bases 5' to 3'. So I'm going to draw it right here. This is DNA polymerase. And this is so far, this is all that it would have had been able to copy. It would just be, it would be right here. Like it would have made, if that's an A, that would put a T down, and then it would be heading toward this place where the DNA is separated. We call it the replication fork. Okay. Now I'm going to draw another piece of DNA right here. And this one, obviously I'm not drawing as neatly. This one is going to have more of the replication fork, or more of the DNA unwound and unzipped. Like this. Okay, so it's the same picture. It's just that now, instead of just having this little tiny piece being finished, I'm gonna put helicase here. I'm gonna put the 3' and the 5' on there. That. And let's go back and get, I think I used this one for helicase, my line picker. So I'm gonna put my helicase here. Green. This is really hard to do. I'm getting better. Yeah. So that's the same as up here. That's the DNA helicase. Right. Single-stranded binding proteins are still there. Keep those brown. I'll make them a little thicker this time, right? So they're here, here, and they usually are in pairs, which is why we draw them this way. All right, so I'll put those there, make those a little thicker. And now primase is going to be finished copying all of this, right? So it would have made A with T, G with C, A with T, G with C. And now the primase is sitting here. Or sorry, polymerase. And this particular polymerase, this is when your book starts saying things like this is called DNA polymerase 3. Because there are different polymerases that are involved in replication. So this one, I'm just going to abbreviate it like this, DNA. We just shorten it. We call it POL3, P-O-L. Paul, and then Roman numerals, though. Okay. So that's great. And if I were to draw this a third time, right? Wait a minute. So this third time, okay, got really wide. This third time, okay, why is it a different color? Third time, okay, for whatever reason, it's gray, would look more like this, where there would be very little left to still replicate. And the enzymes keep sliding. So in this picture, helicase is here, still at the replication fork, right? That's still where helicase is. And DNA polymerase now would have finished all of this. This piece of DNA, the new one, the red one that I'm drawing, we call this continuous, 5' to 3'. So I'm going to put the arrow toward the replication fork again, and I'm going to draw DNA polymerase 3 right here. This strand we call the leading strand because it's made in a single piece. Okay. The complementary strand, this strand here that I haven't made a copy of yet, that strand is a lot more difficult to actually copy. You guys hear that? Looks like they're picking up my sticks today. Township is coming to clean up brush. So the complementary strand here, I'm not going to try to draw it on this top one because it's so small, but the complementary strand is made a different way. It's made starting at the replication fork, and the enzymes move out away from the replication fork. And this would be step 4. The thing I didn't tell you about polymerase 3, but you can read on your handout, is when it adds those complementary bases, it does it much faster than I just did. It puts in about 600 to 1,000 bases a second. It proofreads, so if it makes a mistake, it goes in, cuts it out, and puts the correct base in. But remember, it can only go in this direction, 5' to 3'. So it's different for this bottom strand, because for the complementary strand, because it needs to be made in the opposite direction. I can't start here. If you guys can see where my pencil is, I can't put polymerase here because I would have 5', and it's got to be complementary and anti-parallel. This has to end up being a 3' end. So what happens is there's another molecule involved, and I'll see if we can make it pink and see if it shows, right? So what happens is, like I said, I don't have enough space here. Let's do it here on this one. At the replication fork, another enzyme comes in, and I'm going to try to make it look kind of like a triangle. And this enzyme's name is called primase. And what primase does is pink for primase, that'll help us remember. Primase. P-R-I-M-A-S-E. And what primase does is it produces a short piece of RNA that is complementary to the DNA. So like I said, I'm going to make it pink. So it's like DNA, but it's not. It has different sugar in it. And it has U's, uracil, instead of T's. There's no T's in RNA. And it is made 5' to 3'. And that's the important bit. This 3' hydroxyl that's here, that 3' hydroxyl is going to serve as a recognition signal for our friend DNA polymerase 3. So then polymerase 3 is going to see that. It's going to sit down, just like before. Get a fatter line here. And it's going to make new bases, 5' to 3', like that. The primer's still there. Okay, that's going to get taken out at the end. So this makes new bases, 5' to 3'. And in my picture, you can't see it very well because there's only like 10 bases, but this is what we call an Okazaki fragment. Okay. Black and just try to write that. Okazaki fragment. O-K-A-Z-A-K-I. And normally they're about 200 base pairs in length. They're a lot bigger than what I've drawn here. Okay, so let's call that one fragment A for the first one. All right. So I'll just keep doing it with this hand. Okay, now I'm going to redraw this so we can make a second Okazaki fragment, because what happens is as helicase keeps moving along here and unwinding and unzipping, more DNA is going to be exposed, and primase is going to sit down here at the replication fork, make another primer. DNA polymerase 3 is going to recognize that and make another Okazaki fragment. And then we just have two more things to do. One is to get rid of that primer, and the second thing is to join the fragments. So let's see if we can't draw that a little better here. Okay, so this would be my first piece that's already finished. Put it here like that, right? And remember, it still has this pink portion at the beginning of it from the primer. And the primer would sit down here again, and it would make a small primer, 5' to 3', and then DNA polymerase would grab onto that here and make a second Okazaki fragment to right there. Okay, 5' to 3', like that. Okay. Now, the last two steps, that we call this the lagging strand because it's being made in pieces. It's not being made continuously. And the last two steps are to get rid of the primer, which happens by another DNA polymerase enzyme, which is called DNA polymerase 1. So let's see how I can draw that guy. Make him kind of skinny, like this. And we'll label him an arrow. He is DNA polymerase Roman numeral 1. And that enzyme's job is to get rid of the primer and replace it with DNA bases. So that's what it would do. It would put in the correct complementary bases so that we would get this completed fragment. Okay, so this is the same as our first fragment that we had originally. This is the second fragment that we've made, B. And then the last step is to join the two fragments together once they're all made into DNA. And that enzyme, I'll try to draw it. I always usually make it look like kind of like a blue diamond. That enzyme's name is DNA ligase. So let's make that a little skinnier. Ligase. Like to ligate, like a ligament to join together, right? Ligase. So leading strand is made continuously. Lagging strand is made in pieces. The pieces are called Okazaki fragments. Polymerase 1 gets rid of the primer, fills it in with DNA, and then ligase joins the pieces. Now, this would happen again, this combination here of getting rid of the primer, filling it in with DNA, and joining the pieces together would happen again as this opens up and we get more. We'd have another primer sit down, make a little bit of primase would make a little bit of primer, polymerase would join on, make a third fragment, and then polymerase 1 would come in, get rid of the primer, and ligase would join the pieces. So it's made almost, you can think of it, it's made almost like backwards in chunks. The key thing for you guys to remember is it keeps repeating at the replication fork, and it only goes in one direction, 5' to 3'. Okay, I am going to try to, there we